The overall goal of this procedure is to induce precisely controlled damage to zebrafish embryonic skeletal muscle in order to study muscle regeneration processes. This is accomplished by first genetically labeling single skeletal muscle cells in a mosaic fashion. Next, selected embryos are embedded in agarose supplied with tricaine on a slide. Then, the embryos are placed under the microscope and targeted muscle cells are injured using a micropoint laser. Finally, the embryos are removed from the agarose and allowed to recover for defined periods of time. Ultimately, results can be obtained that show changes in myofibrillar organization through laser injury followed by confocal microscopy.
The method that we present today is relevant to the field of muscle regeneration, but can also be applied to other paradigms of regeneration, such as fin regeneration in zebrafish.
To label cells, inject one-cell stage embryos with a plasmid encoding GFP or any GFP fusion protein under the control of ubiquitous or cell-type specific promoter. During development, GFP is then expressed in a mosaic fashion within tissues. In this example, the plasmid injected causes single muscle cells to express alpha-actinin GFP under the control of the beta-actin promoter. Incubate the embryos until 28 hours post-fertilization. Next,